Effects of insulin can be described in two ways: the stimulation of glucose utilization and inhibition of gluconeogenesis, which is the formation of glucose from the non-carbohydrate precursors. In an overall picture, insulin affects the cell membrane, adipose tissue, and the muscles. Insulin facilitates the transport of glucose across the cell membrane from the blood to the tissue. There are few exceptions to this that are liver, CNS, and erythrocytes. The lipid metabolism of adipose tissue is also influenced by this hormone. Insulin stimulates the reorganization of glucose into fatty acids. It also inhibits the degradation of fat by hormone-sensitive lipases. It prevents the breakdown of muscle proteins. In a case where insulin might get deficient in the body, what happens then? Increased glucose concentration in the blood from 5 mm to 9 millimoles may result in hyperglycemia. In muscles and adipose tissues, which are the two most important glucose consumers, over here the uptake and utilization of glucose is impaired. There is increased muscle and fat breakdown in the body. This leads to the stimulation of gluconeogenesis. Glucose utilization in the liver is also reduced. This increases the blood sugar level still further. When the capacity of the kidney to reabsorb glucose is exceeded, glucose is excreted in the urine, causing glucosuria. The increased degradation of fat that occurs in the insulin deficiency also has serious effects. Some of the fatty acids that accumulate in the large quantities are taken up by the liver and used for the lipoprotein synthesis that lead to hyperlipidemia, and the rest are broken down into the acetyl-CoA. If you remember that acetyl-CoA is normally utilized in the TCA cycle. However, in this case of acetyl-CoA excess form ketone bodies and lead to ketoacidosis or ketonuria. Ketoacidosis is also known as metabolic acidosis.